Thank you very much, Alyssa, and thank you all for joining us today. This project is really very near and dear to the hearts of a lot of us who are working on native plant restoration, because part of the reason that we work on native plant restoration are, the challenges the pollinators are, facing from a lot of factors: loss of habitat, climate change. But as we do that restoration work, we need to be thinking ahead to the next challenges, and changing climatic conditions is obviously going to be a big driver of what happens with a lot of our restoration work ultimately. So it's been really great to be able to be a part of this project, Time to Restore, to be able to create the tools that all of us who are out there, boots in the ground, will need to do this restoration work. So as we go further into it, we are asking, in some ways, a lot to have people compile this data, and some of it is challenging, but it will also provide the true backbone for what happens next and what we will be able to do to be able to minimize biodiversity loss, to be able to expand the scope of successful habitat restoration. So the data that you will be collecting through this process is really, really key to all of us and to all of you as well. So to be able to come together and share this, it's a really, really powerful thing. So that's where I wanted to kick things off, just to have context for what the importance and the larger significance, not just now, but in the future, for the work that we'll be doing in this project as we gather this information to give people the tools they need to make better decisions about habitat restoration with native plants. So with that, I will turn it over back to, I think, Erin. Maybe Alyssa. I'm going to turn it back to Alyssa.
Perfect. Thank you. Thanks. So yes, I think Erin will share the slides. Yeah, as Jane said, we're really excited to be here with you all, and I just appreciate the teamwork that we've seen already in terms of the contributions that everyone made to helping shape this project so far through our stakeholder workshops in November. So our focus today is on how we're actually going to go about collecting this data on life cycle events in plants in order to develop the information on bloom and seed timing and how those are going to be affected by climate change. And so we're going to be using two platforms, iNaturalist and Nature's Notebook. And I'm going to start by describing a bit of, I'm going to start with iNaturalist. So if you want to pass to the next slide. So iNaturalist is a really wonderful and easy to use program for collecting presence data on plants and timing of flowering. There's a mobile app, if folks haven't used it already, probably many of you have. There's an easy mobile application where you generally submit an observation with a photo and a georeferenced location. You say whether it's captive or cultivated, and then all of that data is available for download and analysis, and you can also look through recent observations and see the seasonality. One of the main differences between the two platforms, between iNaturalist and Nature's Notebook, is that in Nature's Notebook you pick an individual plant or patch of plants and you monitor the same ones over time, so you know what that individual was doing in March and what that individual was doing in April. Whereas iNaturalist is more opportunistic, more just, this plant on this day, the record is kind of self-contained. It's this plant was in flower on this day and you're not necessarily going to return to it. So it's awesome for species identification, for one-time observations, like with casual park visitors who aren't going to engage again and again and again over time, or for even yourself visiting a place you won't regularly return to, but you saw, Monarda with seeds, and so you're going to put that there. And awesome, thank you, Annie, for sharing the app link. And then I wanted to invite Brandon or Colin, if they're here, with Tribal Alliance for Pollinators, to share a little bit about their experience with iNaturalist, since I know they both do a terrific job using it.
Yeah, iNat is super useful. We use it like basically every day. It just has so many options that you can use on it, and you can see fungi, you can see plants, you can see birds, everything that occurs in Oklahoma. You can see it, or anywhere in the world, really. You can see it and post about it. You can get research grade people a lot of times for plants. It's Dr. Amy Butode. She's IDing thousands of plants on there. She's the top by far. And you can see how many observations there are. So some species are going to be more rare inherently than others, and you can kind of see that distribution. And it's really useful for seeing, so for example, Liatris picanastatia, or prairie blazing star, basically stops in the middle of the US. It's very prevalent on the east side, but the west side, it basically doesn't really occur except in like California. But yeah, it's really, really useful. Brandon, you got anything to say?
Yeah, I would add on to it. I took some botany courses in 2014 to 16 era, and the apps were around then. They were just sort of getting started, and man, they were just pretty much useless, entirely useless. And so I learned sort of by using keys and stuff like that. But nowadays, you use these apps now. It's incredible how drastically they've improved just in such a short amount of time, and they're constantly getting better and better. I use it quite a bit just to sort of give me a ballpark area. I wouldn't always trust it for a species level, but for a genus level, it's usually pretty good, or it will at least get you some similar family nearby, and then you can kind of explore a bit that way. So yeah, I definitely recommend it. It's made botany and identification way more accessible, in my opinion. It's very easy to give to beginners.
Awesome. Thank you both so much. And we'll have another moment at the end if people have more questions. I think because of your deeper knowledge, we'll point people back to you both. So yeah, Erin, if you could kick to the next slide. So through these workshops that many of you attended in November, we honed in on a list of 8 top species that we're going to focus on across the region. And just as a reminder, the region, or you may have, is New Mexico, Texas, Oklahoma, and Louisiana. And so we're going to aim to be observing these 8, which were like got the highest number of votes across the region. But we understand that there'll be species of interest to you, or species that are more accessible, that'll like make sense for your location and your implementation. So there's a much longer list, and that we'll be sharing in the follow-ups. But these 8, wild bergamot, cardinal flower, common sunflower, showy milkweed, tall blazing star, eastern purple coneflower, buttonbush, and green antelope horn are the primary species that we'll be focusing on for this project. And so now I think it is over to Gail Bishop with the Gulf Coast Phenology Trail.
Good afternoon, everybody. I'm glad to be with you. I have been working as a trail coordinator for about 5 years with the Gulf Coast Phenology Trail. So I just wanted to have a few slides to share with you. Some people want to know, well, where can I observe? I know when we had our workshop with the folks in Louisiana, some of them might live in a small location. They've got their plants on patio pots. So really you can observe everywhere. I know from experience leading some of the training, a lot of our participants didn't necessarily want to go out and be part of a group, but just wanted to know about their own backyard gardens and when things might bloom, and to have some kind of setup for that. So thank you. Next. But you can also do it at group sites. We have 11 sites in the Gulf Coast Phenology Trail right now, but this is a site that I've visited several times. It's a butterfly garden at the Anahuac National Wildlife Refuge in southeast Texas. And what's nice about this location is that a lot of the plants are already identified, but it's very helpful to go in a group site. Next. And people want to know, well, can I just go out and start observing? But you really need to have the training, but it's really helpful to observe with a partner. Sometimes when you first get started, just not really clear on, well, what am I looking at? I had an example When I first started observing, I looked at Yopon holly, and the little tiny berries that start forming also look like a plant gall. And I would often get that confused until I figured out what it was. So that's really helpful. The next slide we have is about looking at plants that are either individual flowers or patches of flowers. I know buttonbush you want to observe as an individual because it has multiple flowers on a shrub, whereas you might find some places are you've got multiple flowers, or you want to establish a patch. Now that's a little tricky. You have to decide how much you want to cover in a patch. When I've observed at the Sandhill Crane Refuge, I looked at pitcher plants, and I would just have it one place that I go back to over and over again. And that's what you want to do. You want to keep going back and looking at the same location to see what's coming up. But when I had that one, I used a landmark, and my landmark there was just a wayside exhibit. And then I visualized how many feet or how many square feet that I was going to observe. So those are your options. Next. Now there's some annuals that will die back, sunflowers to seeds. So here is an example of a sunflower, which is one of the ones we've got listed for our focal plants. And so just know that these will die back. So it's good to collect the seeds when it's appropriate. I think you've got a slide there also of seeds. So we'll go to the next one. And animal observations, we're focusing right now just on plants, but certainly when you're out and you see, for example, this monarch butterfly on the blazing star, I like recording that, but you want to have your animals already listed in Nature's Notebook if that's what you want to do. So it's really good. We're really interested in monarch butterflies and also Eastern Baccharis because they do use that plant. So it's really helpful to see which plants they're using for nectar. So I have one more or several more, and this is what I use is Nature's Notebook is a free app that is very helpful. It has the standard protocol, what questions and how to answer them, yes or no. And if you have a question, well, I'm not really sure. If you can kind of see it on the screen, they'll have a little icon, and it goes into a lot more detail. If you're questioning, well, I'm not really sure, I'm not sure what I'm looking at, or if it's got a dead leaf or a dried leaf. So it goes into a lot more information. Now, this might seem overwhelming, but I find it extremely fun to use. I like using the app on my phone, but there is another way to do that, and that'll be on the next slide. And that's paper copies. Now you can go to Nature's Notebook and go to when you set up your observation deck, there will be specific paper copies that you can use. The problem with this, then you've got to do the work twice. So you would go out in the field and you'd answer that, does this plant have young leaves? Yes or no leaves. And so you'd go down that pick list and then record it on your paper. Well, then you got to go back to your house and find on your personal computer and go to your website. Then you've got to do it again. The other disadvantage I have for that is it does not have that opportunity to go into detail unless you bring the papers with you. So you've got more to carry. So this is not my preference, but it is an option if you don't have access to a computer when you're observing. We have another slide. And this is some of the resources that are available to you. Training resources. This is one of the best classes is if you go to the learning, you can see at the bottom of the slide there, it's got the information how to get to that site. But this is a great module, especially module 1, just to get you started on looking at what am I getting into? What am I going to be looking at? And so this is a really helpful plan, is to take the training online at your own, on speed, and just enjoy looking at that. So that's it. Now, some other people have been making their own. This is a phenology project, and this is called, this is wild bergamot, which is one of our focal species. So it has a lot of great information. The description, where you might find it, the phenology highlights, the facts about it. I've made a few that are, they're still in progress because sometimes we don't have all the photographs of that particular plant that are needed. But another group, and that was at the Jean Lafitte National Jean Lafitte National Historical Park and Preserve, which is south of New Orleans. They were able to get an intern to go and look at all of their plants, and over time, or she was able to locate additional photographs, they made their own plant guide. So that's really helpful. So you've got many options. But the one thing that I'll highlight, and I'll say it over, is that if you go to the Nature's Notebook or Nature's Notebook web page, there is just an incredible amount of information that will help you as you kind of get started on the project. So I recommend that, and that's what I would like to leave you with, is go on the website and use the information that's available, because there's so much. Thanks.
Thanks, Gail, for that great overview. So I'm going to get a little bit more into detail about how you can do this in Nature's Notebook and give you some more of the specifics on how you can set up a monitoring site and add different plants and things like that. So the way that Nature's Notebook works that is different than iNaturalist is we have a set location where you do your observations repeatedly over time, over the long term. So when you create an account in Nature's Notebook, you're asked to set up an observation site. And so the considerations for that are, you want to make sure that your site is convenient. You know, we don't want you to pick the perfect site that might be the most representative of all the different areas around you, but it's really hard to get to, and then you'll never go there. So we'd prefer you find something that's, you know, convenient to you, where you can get to it maybe once a week if possible. And if you can find something that's convenient, think about, try to make it representative of the ecosystem that's around you. We also recommend that if you can, to try to make it be kind of a uniform habitat. So we have this little diagram here that shows, you know, if you're in an area that had really different parts, different forest types, maybe a deciduous forest type and then evergreen, maybe there's like an open meadow area, you might consider setting that up as different sites. You can have more than one, but just try to remember that a site is supposed to be kind of a representative area that's near you, that when you collect the phenology data at that location, you're kind of reporting what's going on at that place. And so it should be fairly consistent. And then in terms of the size, it really depends on what you want to observe. So with this project, you know, we're looking at nectar plants. You can have it be one nectar plant or a patch of plants, whatever is kind of convenient. It could be your backyard, or it could be a nearby park that you like to go to. A public area, and then make it something that is not too large that, you know, you're kind of going beyond the boundaries of what you're looking at with your plants. Make sure that it encompasses the plants that you're observing, but beyond that, it can be kind of any size that you like. So thinking about how you would go about selecting plants, I just wanted to go through kind of an example of how you might do this. So let's say you have kind of a meadow area that has a few different species on it, and some are more individual kind of plants, and some are in patches where you can't really tell apart the individual stems. In this example, you might decide, I'm going to pick 2 different plants here, and I'm not sure which species this is supposed to be, but let's call them penstemon. So 2 different penstemon plants, and then 1 patch of poppies. And so when you register this in Nature's Notebook, the way you're going to do it is you're going to register penstemon number 1, penstemon number 2, and then poppy patch number 1. And so you have 3 different plants, individual plants or patches that you are going back to observe each time you make a visit. And we also recommend marking your patches and plants just so you can find them again. So if you can put a little flag or just maybe take a picture of it, you can upload a single photo of each plant or patch in Nature's Notebook to help you find it again. Just something so that you know where to go back to. We've had some people make Google Maps and then mark on there where their different plants are, but just something that you can remember that you want to go back to the same individual plants or patches each time you make a visit. And then in terms of frequency and monitoring, for these species, we have kind of a general sense of when they're blooming. So I put that on here in this table. It's going to be really different depending on where you are in the region. So don't take this as this is definite. One of the things we're trying to do with this project is get a better idea of the timing of flowering. But just to give you kind of a general sense, if you pick something like sunflower, you might be monitoring anytime during the year because those can flower pretty much all year, depending on where you are. But if you pick something that has a smaller or shorter period, you won't really have to worry about that until later towards the summer. And then you might be finished with monitoring that. So it's kind of up to you, depending on what species you have near you, how much of the year you might be doing the monitoring. But we're really interested in the flowering and the seed timing for these species. So in terms of how often you should monitor, we would love it if you can monitor once a week or even more frequently at the times when things are really happening. What we're really trying to get information about is when flowering is starting and when the peak in flowering is happening, and the same with fruiting, so or seed timing. So it's really important if you're going to do more observation effort, if you try to target those times, right before things start opening, and then right when things are really starting to peak and you have the most flowers open, you might want to go more frequently. But we would love any observations that you can do. So if you feel like you can't commit to more than, once every couple weeks, that's totally fine. We would still love to have your observations. So the data sheet for something like a milkweed or another forb looks like this. So it's pretty simple. We have a couple questions about leaves that are not really critical with this project, although you're welcome to report on all of the questions. But the ones that we're very interested in are the open flowers and the ripe fruits. So those are the two that we're really emphasizing here. And so each time you make a visit, you're going to be saying either, yes, I see open flowers, or no, I don't. Or if you're not sure, you can always use the question mark. And then you'll see there's this little line here next to the question mark, and that is for the intensity. So we have a second question in Nature's Notebook that's about how much did you see? So for open flowers, we're asking you what percentage of all the flowers are open? And then we're reminding you here that some of the flowers that we're looking at here are in inflorescences, but you're really going to try to estimate the percentage of all of the little flowers that are open. If you want to report on the flowers or flower buds as well, that one is actually just looking for any flowers or flower buds that are on the plant. So if they're open or if they're not open, we want to know if you see any flowers or buds. And the question for that one has to do with how many flowers are there. So if you are looking at a species that has inflorescences, like a spike of flowers or an umbel or something like that, you're actually counting the number of those inflorescences on the plant or on the patch. And we'll have some pictures to try to practice this in a minute. And then for fruits, it's similar. If you want to count the fruits, you could do that and count how many individual fruits you see, estimating in these bins. And then with the ripe fruits, we're looking at what percentage of those fruits on the plant are ripe. And so as Gail was mentioning, we do have all of this information on the app. It's that little I that's next to the question, so you can always access that information when you're out in the field. And then if you're using the paper data sheets, you can choose to print out this definition sheet as well and take that with you. So just to kind of emphasize with the monitoring frequency, why doing more observations might be more helpful. We're really trying to pinpoint things like when flowering starts. So in that case, it's really great if we can have a few number of days in between the last time you said no to open flowers and then the first time you said yes. So in this example, it shows if you're only monitoring about once a week, we know that there's about 6 days in between the last time you said no and then when you said yes. So the actual time that that plant started having open flowers could have been any time in those 6 days. Whereas if you're going a couple times a week, we can narrow down that window to just 3 days. So if you're going to try to increase frequency at all, it's really helpful to do that around the start of open flowers timing and then when that peak is happening as well. So again, we are looking at these two kind of pieces. We're really interested in the start of flowering and the peak. So this is another just kind of conceptual graph here that shows what we're looking for with the peak and open flowers. If you're reporting on these different percentages each time you visit, and you're gradually showing that there's a greater and greater percentage, we can really identify when the most flowers were open on the plant, and that's really useful for identifying that peak in timing. So I have a couple of pictures here to kind of help us practice how this might work if you're out in the field. So here, this is wild bergamot, and we are looking for the question about, do you see flowers or flower buds? And so hopefully you can see this. I might need to move my Zoom panel out of the way. Let's see. Okay. So you can always refer back to these definitions to make sure you know what we're asking for. So here with flowers or flower buds, we're asking, do you see one or more fresh opened or unopened flowers or flower buds? And it reminds you that some of these plants occur in inflorescences. So in this case, we have a bunch of little tiny flowers in a flower head here, or an inflorescence. So if we were going to try to count how many flowers or flower buds are in this patch of plants here, you might try to just identify a part of the plant where you can easily count how many inflorescences or flower heads. So maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. It's 10 or so. And then you can take this area and see how many of those sized areas are on the patch and multiply and try to kind of get a sense. So I would say there's probably at least 10 of these circles on here, which is going to put us well over 100. But probably not more than 1,000. So just remember that we're trying to get an estimate, and we have these bins to help you. So if you were going to estimate, I would put this in the 100 to 1,000 range, and I think that's a pretty safe estimate for this plant. So the next question that you get asked is, do you see open flowers? And again, if we go to the definition, what we're looking for are the flowers are considered open when the reproductive parts are visible. And so we want to make sure we can see the reproductive parts, which we can. There's a lot of them here. So we can definitely say yes for this question here. And then in terms of the percentage that are open, you're considering how many of these individual little flowers are open. And you might have a situation where the flowers might be different from plant to plant, or from flower head to flower head. So you might have, in this case, all of them are open pretty much. You can see all the reproductive parts. So I would put this in the 95% or more. So it's pretty consistent across flower heads, but you should just kind of take the average of your plant or your patch in terms of what percent of these little flowers are open. So we have one more example here with a button bush. So in this case, this is more of a shrub. So this is going to be an individual plant that you're monitoring rather than a patch. So with this, we would be looking at the same question again about flowers or flower buds. And again, you can kind of pick an area on the plant and then extrapolate from there. This might be maybe similar to the last one, where I think there's probably at least 10, but probably less than 20 flowers in this. So it would probably be again in that 100 to 1,000, but you could also, we could do this and, multiply out by the same size shape across the whole plant. Some people in the field will do this with a clipboard or holding up your hand and kind of covering up part of the plant and seeing how many flower heads are behind your hand, and then multiplying by the number of hands that you can cover the plant with. So there are different ways of kind of estimating that way. And then if we look at the open flowers, so again, we're looking for the reproductive parts. And in this situation here, in this example, they're not all the same, right? There's some that are totally open. So this flower head is 100% open, but there's some that are closed. So if I was going to take the average, I would say, well, I have 1, 2, 3, 4, 5 different flower heads. 2 of them are 100% open, but that's still less than half of my overall little flowers. So I would put this in the 25 to 49% category, because it's just less than half of my total flowers that are open. And thinking about it in terms of the pollinators too, it's how much nectar is available on this plant. So what percent of the potential flowers are open and with having available nectar and pollen for these pollinators. And then we have one example, I think, for the fruits. So in this case, we are looking for how many fruits we see on the plant or on the patch of plants. So with this milkweed, it's kind of, you might not know exactly what does count as a fruit on the species, but our definitions have what we call species specific definitions for each species that we have. So it'll tell you exactly what you're looking for here. Oops. So if you look up here, I can't quite see it because my Zoom is in the way. Let's see if I can move it over. Oh, there we go. So for this species, the fruit is large and pod-like and changes from green to tan or brown and splits open to expose seeds with fluff. Do not include empty fruits that have already dropped all of their seeds. So we're pretty sure looking at this is green, so it's probably unripe fruits here. And this is one fruit, and this is another fruit and another one. So it's fairly easy to count these. So in this case, I would say I see fruits, but I don't see ripe fruits. And I would say for the count, at least in this picture, it's going to be in the 3 to 10. But if you look at the whole plant or patch, you might, count, and it might be above 10 because we're just looking at a small part of this plant. So fruits are a little bit more straightforward. You're just counting. You're not counting the inflorescence. You're counting each individual pod. And then I also wanted to talk about an example where it might be very difficult to count the fruits. And so in this case, we're looking at, for this plant, they're super tiny little seeds. And so don't worry about trying to count these, with these species where it's really hard. If you just can answer yes or no for these questions about fruits and ripe fruits, that's really helpful information. Don't stress about trying to count, or see what percentage of these are ripe. It's going to be really difficult, and you might be really messing with the plant to try to get all the seeds out. So you can just report those yes and no answers for these kind of species. And I also wanted to mention that this project, Time to Restore, is linked up with one of our campaigns in Nature's Notebook, which is called Nectar Connectors. And so our campaigns basically are a way to ask you to report on particular species that are of interest to natural resource managers or scientists, and then give you results back about what we're finding. So when you sign up for messages as part of this campaign, we will send you approximately monthly messages that have lots of information about identification tips for the plants and the life cycle stages, and lots of results about what people are reporting. And so this is a great way to kind of stay in touch with us about what people are finding, how much data has been collected on different species. And so I encourage you to sign up for the campaign. We'll put the link in the chat, and there'll be on the right sidebar of the page, there's a link to sign up for the messages. And so you'll just get messages, as I said, like every month or so. And that'll really help you see the data as they come in this year. All right, I will pass it over to Alyssa for this part.
Thanks. So yes, we realized that it was probably going to be kind of a lot to get into an hour, but we wanted to at least give you a picture of the approach that we're thinking about for gathering data on these species to inform our questions. And I think from here you have a good intro to iNaturalist and Nature's Notebook, and you could maybe before hosting your virtual and training program, plan out what the effort might look like for your organization in your state. And then think about maybe a virtual or in-person or a mix of both in terms of training other people. And then as Gail just talked about this, there's sort of two ways it can run, and they're both great, right? Like one is setting people up to observe in their own backyards or local parks, where each individual person is tracking individual plants, or group sites where people cooperate to, that works really well, like I live outside of Boston, and here at the Arnold Arboretum, they have this schedule where a different pair of people observe each week so that they're getting that consistent data, but the people are teaming up to collect it. So you could have individual or group sites for your effort. And then also it's a huge help to the overall project to help us get the word out, help recruit new people to participate and connect with us. And so I wanted to also then highlight our state coordinators who are on the call with us today. In New Mexico, Kim Eichhurst with the Bosque Ecosystem Monitoring Program. In Oklahoma, Jane Breckenridge, as well as April Taylor. And then in Louisiana, Gail Bishop and Sue Wilder. And so they're co-leads on the project and can provide inspiration and support and resources to you as well. And so I think from here, we were going to open it up for Kim, Jane, or Gail to share how their efforts will look this year, and anything else they'd like to chime in at this time.
Thanks, Alyssa. I'll just jump in really quickly. So here in New Mexico, of course, not all of the species that were highlighted across the states are what we're going to find, especially in our native areas. So we're focusing primarily on what some of our partners are wanting to focus on when they're looking at restoration projects. So we'll have about, I think, 3 or 4 of the key species, in addition to other species of interest for like the City of Albuquerque restoration projects. So we're looking at smaller in-person trainings that are outdoors and following COVID-safe protocols, but certainly we're interested in more online trainings if that is of use for our partners across states.
Right, well, I'll jump in. So right now we are still just kind of figuring out with the COVID situation, what our training schedule is going to be like. I guess everybody's in this same kind of situation. However, it is our goal to plan for this spring to have hopefully an in-person training workshop at the home base of the Tribal Alliance for Pollinators, which is the Uchi Butterfly Farm just outside of Bixby. We've got a 13-acre prairie remnant there that is a good site to do training as well. But we'd also like for all of the people that are participating in Oklahoma to get feedback from you about what would be most useful for you as we move forward with training and learning how to, you know, this is a train the trainer meeting. So as you get farther into it, what we can do to help you as you bring your different partners and participants in. So to that end, please feel free to reach out to me. You can reach me at jane@nativebutterflies.org, or you can reach out to Colin or Brandon, who are coordinators for Tribal Alliance for Pollinators. And just, you know, we welcome your feedback about how this works best for you as we kind of work our way out of this COVID situation. But in theory, we are planning to do in-person training workshops outside at the farm this spring and summer. So. And there's Brandon has put his email address there in the chat box. And and if even beyond this project, any way that we can be useful as you work on your native plant restoration and your pollinator projects, we're very happy to do that. That's our mission. So please feel free again to reach out to me or Colin or Brandon.
Hi, everybody. This is Gail again. I just want to let you know that we've got some things kind of in the pipeline as well, kind of wait and see what's going to happen with COVID in Louisiana. But I will be meeting with the New Orleans, it's the City of New Orleans Native Plant Society this weekend. We have a weekend of events planned, and I'll be speaking there and hoping to meet up with folks and kind of get their idea what they would like to do with the training. And plus, you already have some sites in Louisiana that we'll get with the managers and talk about this new addition to Gulf Coast Phenology Trail. So that's kind of what we've got right now. And I put my email address, that's my personal, but I'll also put gulfcoastphenologytrail.com in another message. So you have two ways of reaching me, but I really look forward to this project. Louisiana's got some great locations to observe, and I know their folks are really excited about this. So thanks.
Awesome. Thanks, guys. Great to hear about all the plans coming up. And definitely, we want to hear from all of you about any questions that you have about the project and what we've talked about today, and whether you are interested in doing a data collection training, and we can definitely talk with you about what you want to do there. I did just want to leave too with some things that are coming up in the next couple months for the project. So in the next probably few days, we will send you a bunch of materials as a follow-up. So we'll send you this slide deck from today. We'll send you a couple handouts, one that Kim compiled for us that has kind of why we're doing the project and how the data collection fits into the project that you can use. We'll also have a handout that focuses on those monitoring guidelines for how to select a site and register different individual plants or patches. And then some other training materials that are on the different websites as well. And then in March, we're hoping to have a check-in meeting just to touch back with you and see how it's going. You know, if you have any questions or need any help with anything that you're trying to do with the data collection. And then over the summer, we'll have some preliminary results to share with you. We'll show you what everyone's collected and see where we're at with the data collection. So let's see what's in the chat. If there's any questions there, or feel free to unmute yourself if you like and ask any questions that you have. So no questions yet, but let's see. Does anyone, or is there anything in particular that you feel like you would want to have in the follow-up in terms of like more detail about anything that we've talked about, further instructions or more resources or anything like that?
Yeah, Jacqueline mentions funding resources. Feel free to unmute. It's sort of, that is something that we sometimes share around in newsletters, but just sort of knowing the scale and scope and what kinds of funding you might be interested in could help.
Yeah, Jacqueline, who are you with? What organization are you with?
Hi, can you guys hear me?
Yes.
Oh, good. Okay. I'm with Delaware Nation, located in Anadarko, Oklahoma. We're kind of central southwest in Oklahoma. And well, I think I've actually spoke with you, Jane, briefly before COVID about starting in pollinator plots. And we're trying to get an apiary and an orchard going and a community garden. And so we have this big view, but we're having problems getting off the ground. We have the ideas and we have the technical people like you guys who we can reach out to. What we're running into is finding the funding to start the projects. And so I did apply for the Native American Agricultural Fund grant. Unfortunately, because it's so competitive, we didn't get it. So we're just going to keep trying. But so some things like that will cover maybe starting the plots, the areas, getting some good compost in because our soil is really sandy. It's sandy loam. And so sometimes I know our native plants will do great in it. So we may not need a soil amendment on that. But we're just looking for any funding that could really get our ideas off the ground.
Okay. How many acres do you have there at your tribal complex or on your tribal lands?
As of right now, for the community garden area, we have 2 acres, and I've been approved to use the surrounding community garden with as pollinator plots. And also I can do plots within our area. So the area isn't just a community garden. It has it'll have ADA compliant swings It'll have like a few sensory gardens. And so it's kind of just like a big community center. And then we've just been approved to use an acre of a forested area next to our child care center. And that is 1 acre, and it's next to a seasonal pond. It's really mostly just like rainwater runoff. But I've been talking with our tribal administrator, and she said that we could, get some funding, stock it, get it going, so then we can possibly use it as something to draw the water from for the orchard. And so we have big plans.
Yeah, no, yeah, they're beautiful and brilliant plans. So we could send Colin and Brandon down to take a look at your site. Do you have any other larger acreage that you control as a tribe besides those acres that you just described around the complex?
I believe we do. I don't within my program, but I know we have to the back of our complex, our headquarters, there's some acreage that our tribe and the Wichita and Caddos, I believe it's a shared area of land, but I'm not sure on the amount of that. I would have to get with my tribal administrator and see, but I know we could probably use what's around the complex, and that's probably at least a ½ acre of just where people don't walk. It's just kind of just like an area that used to be grazing, and now they just fenced it off, and it's part of our area on our headquarters side.
Okay.
And so right next to us is a grazing field.
Okay, so we, I may have some resources for you, but I'll follow up with you afterwards. Do I have your contact information?
I will email you, and then that way I'll CC Colin and Brandon.
Yeah.
And then
Yeah,
that way
yes.
we can get another email going, because I think Colin's emailed me, but it was pre-COVID, and so we kind of just you know how it goes. All the plans was kind of halted.
I know everything was disrupted. Yeah, we feel the same way, but we do have resources, and this is exactly what our mission is. And so we love the tools and the partnership we have with the Time to Restore folks. But we also have our other mission, just working very hands-on with tribes trying to restore these native plants. And we do have some different funding and resources. I mean, not a huge amount of funding, but we're starting a new project with National Fish and Wildlife Foundation. And so anyway, I'll email you that. And so that may be a possibility for something you all could participate in as well. So,
Great.
yeah, so we're happy to help. Thank you for logging in today, and thank you for your interest in participating in the Time to Restore project. It's really important work, and we're grateful that you're participating.
Thank you, and thank you for all of the information you guys have given us today, and always.
Okay, thank you.
Yeah, and we have also, Erin shared that list of, it's short so far, list of funding opportunities, but we have an eye on continuing to share that, and then just using this group of us as an opportunity to network and find people who might be interested in collaborating. So I think some of our future calls will also have that more explicit chance to find people who might want to collaborate on a project for funding.
Yeah, definitely. Okay.
Other questions?
Thank you.
Comments? Thanks, Jacqueline.
Does anyone feel like you have either a group of volunteers or potential data collectors that you already have in mind? We'd love to hear about any ideas that you have there or plans, so we can help out in whatever way we can. Also, if you feel like you're not quite ready to share, or you're not quite ready to do a training, what would get you there? Is there something else you need, something else we can help you with? Would you want to go in and do a training with one of our state leads? Any kind of feedback would be great. Sorry if I cut you off, Jane.
No, I was just going to say, this is a very quiet group. Our seed collection group, we got so long-winded, we went hours over schedule or something. And this group is so organized, to the point. And anyway.
We probably rushed through the slides way too fast, but we will include the slides in the follow-up email so you can take a look at them more closely. And yeah, just anything else that you're feeling like, oh, I really wish I had some more detail on this one thing, feel free to share.
Hey, Erin. This is Jackie again. The idea of having a hands-on workshop before we put on our own workshop, I would love that. I would go to that.
Cool. Yeah. So like a little bit more in-person train the trainers thing where you get that experience in the field looking at plants.
Yes.
Yeah.
Yes.
Cool.
And that's what we're hoping to do with our Oklahoma workshop, is exactly that, because it's one thing to see it on a slide, but particularly for those of us who are not, botanist or PhD scientist, it's the hands-on component is so important. Just being out there and being with the plants and being with patient teachers. And that's what we are hoping to do in our Oklahoma workshops, which, like I said, hopefully are going to be in person and outdoors in later spring. So. And that's great feedback. Thank you.
I have a question, which might be more of a different or a further training. But I work with Kim Eichhorst at BEMP, and we collect a lot of phenology data here in Albuquerque. And I was wondering if it's possible from the website to download data from specific plants.
Yeah.
There is? Okay.
Yeah. Actually, in both programs. So iNaturalist has a data download tool. And then through Nature's Notebook and the NPN, we have a visualization tool. So you can actually look at the data before you download it and see what's there for different species. And you can restrict what you're looking at to a state or a region, or even smaller locations than that. And then you can download basically Excel files with those data. But yeah, they're all really accessible. And we could put links in the follow-up email too with all that stuff.
Okay, cool. Yeah, and I've used the visualization tool, and it is really cool. Is it possible to even do it like by plant, like within our group? Okay, just like in one of our monitoring plots, I can pick from this plant. We can see everything about what we've collected.
Yeah, so you can't look at an individual plant very easily on the Viz tool, but you can look at, like, if you selected BEMP, the Bosque Ecosystem Monitoring Program, as your group, and then we have on the listing of all of our partners, you can get into the Viz tool with a filter set for just BEMP, and then you could look at all the BEMP data on a particular species, and then you could even like apply a geographical boundary, so you could like restrict even further on the BEMP data. But I don't know if you could get to the point where you would look at a single plant, but you can do at least a phenology calendar on your observation deck, which is like where you're logged in Nature's Notebook. And there you can do an individual plant.
Okay, awesome. That sounds great. Thank you so much.
Yeah.
And once you filter the Viz tool down to that like specific site and partner, Erin, right, you can also hit a button that says download this data, so that you can get an Excel file that is real tight to that thing, and then you should be able to see the individual plant IDs there and delete the ones you don't want.
Yeah, definitely. I also wanted to mention, if you're feeling like you're not really at the point where you want to do data collection yet, we're also just looking for help with getting the word out to potential data collectors that might be what we call our backyard observers. So they might not be as part of an organization, but any individual can participate in either of these programs. So if you have like a channel where you can like spread the word, we'll have information in the follow-up email that you can send out, and just encourage people to either use iNaturalist or Nature's Notebook to collect information about those 8 priority species. And that would be super helpful for just getting more people collecting flowering and seed data on those 8 species. And then the other way you can contribute that would be really helpful is we're looking for good photographs of these particularly the 8 species, but also the larger list, which we're going to share in the email. And so Gail had mentioned we have those guides that we make that show, like for each of the questions on the data sheet, what does it look like in a picture form? And it's really helpful if it's a very high resolution photo that's very zoomed in on what we're looking at. So it might be, showing the reproductive parts on the open flower or showing what the ripe fruits look like. And so if you could get more people to take pictures, we'll also have information about where they can send us those pictures so we can make more of those guides. Awesome. Yeah, the promotion is very helpful. So thank you. Any other questions or thoughts?
I was struck, like, thinking about our presentation today, the iNaturalist and the Nature's Notebook, and just the in-depth that you go into looking so closely at those flowers. Like, it can seem like a burden, but it is also, especially if you're like, just take it slow and step into it. It's this amazing way to like learn and get to know these plants more intimately too, and just really see what those seasonal changes look like close up. So hopefully that has its appeal as well to you and others that you might engage with.
There. And one other thing I'll add is that for our tribal partners out there, and I guess this is true for non-tribal partners too, it's a wonderful thing to get the youth out there and involved in, STEM learning and getting them connected to the plants. So that's what we've seen is for just a lot of our tribal partners, it's been an awesome thing to get the youth involved in this kind of work and gathering that scientific data. And it's just, anyway, plus more labor and getting the different youth involved. So something to think about.
Well, we can hang out here for a bit if anyone wants to chat, and you're welcome. If you have a particular question for any of the state leads, we'll all hang out here for a bit in case you want to chat with us. But I think I will probably end the recording now, and then we will be in touch with you with lots of follow-up information in an email later this week. But thank you so much, everyone, for coming.
Thank you.